This is Selma Schimmel for the group room in San Antonio at the annual San Antonio Breast Cancer Symposium. Happy to be joined by Dr. Edith Perez, Deputy Director at Large, Mayo Clinic Cancer Center in Jacksonville, Florida. Welcome back.
Great to see you again.
Today we're going to talk about some of your research as it relates to biomarkers and how they are used to help predict and guide therapeutic options.
It's a great topic to discuss. Over the last several years, patients, physicians, regulatory agencies have learned that we have made significant advances in the treatment of patients with breast cancer, in the advanced stages and also early stages of breast cancer. But we also know that not all patients benefit from all of the therapies that we have available. So a significant amount of time of my research and the research of my team has been devoted to try to figure out whether we can identify biomarkers in the tumor specimens that may help us predict whether patients have a good likelihood or not so good likelihood to benefit from treatments.
Would you be able to highlight some of the clinical discoveries based on your work and how it's impacting breast cancer?
Yes, one of the areas that I've really concentrated my effort on over the last actually 12 years has been a HER2 subset of patients with breast cancer. As you know, we have participated in many of the clinical trials identifying the benefit of trastuzumab in the adjuvant setting, trastuzumab in the metastatic setting. We are involved in the evaluation of lapatinib in the adjuvant setting. I'm involved in research looking at pertuzumab and T-DM1. We also know that although significant improvements have been made with these agents, not everybody benefits. What we have done is an evaluation of a whole genome, which means evaluation of the gene products of specifically 29,000 what we call gene transcripts in the context of one of the very large adjuvant trastuzumab trials called N9831. So this was one of the trials that helped the FDA approve trastuzumab in the adjuvant setting. So what we did is that with this analysis of gene profiling in these tumor specimens, connected with the clinical outcome data, is that we've been able to identify a subset of genes that are associated with patient outcome. And these data are really revolutionary. Specifically, we found 32 genes that are highly correlated with patient outcome. Of those, 27 of those genes are associated with good clinical response to chemotherapy with trastuzumab, and 5 of those genes are associated with not so good outcome to these therapies. So a question that you may have, and that we all have, is what is the clinical significance of these findings? And what are we planning to do next? Well, in terms of clinical significance, my goal is to be able to help develop an algorithm or a test that physicians will be able to use in the next few years, in which they can take a tumor specimen, perform gene profiling, and tell a patient you have a very high likelihood of benefiting from anti-HER2 therapies, or you have a not so good option, so the patients can get other treatments. But in order to do this, I need to do a little bit more work. So the next steps will be to collaborate with researchers from the United States as well as other parts of the world who have also conducted adjuvant trials evaluating trastuzumab. And I've been very fortunate that I have several colleagues who have agreed to collaborate with us. So we're going to get their tumor specimens. We're going to receive the annotated clinical information. We're going to be able to put our data, connect our data with their data, and move forward.
As you talk about the discovery of more genes that are identifiers, will there still be more? Where does it end? How many more discoveries can there be on this genomic level?
Right now, we know that there are probably about 25,000 genes in our cells. So we look at not only single gene expression, but we look at correlations between different genes. So I think there's still a lot of more work to be done, but it's very good for people to know that discoveries that can have impact on patient care are here today.
Dr. Perez, as we talk about the specimen collection and some of the challenges that you have to face to get quality specimens and enough to do your work, explain to us a bit more about the process and how this is all used in research and the new innovations in the way you are preparing your tissue?
I think the first strategy is to have a vision of what can be accomplished. And our vision is that we should be able to correlate the patient's situation with what happens eventually to the patient. So we can obtain tumor specimens. Sometimes we can obtain blood specimens, and in the blood we can try to evaluate for the presence of proteins that are circulating. We can evaluate for the presence of circulating tumor cells. And we can do molecular studies to determine if by using those other specimens, we can also better explain patient outcome and benefit to different therapies.
And the cost associated with all of this for the patient?
Yeah, we try not to have the patients incur any costs associated with this type of research. So when I develop trials, as part of our team effort, we obtain outside support so that we can actually pay for the collection of the specimens, we can pay for the shipping of the specimens as well as storage of the specimens with an eventual plan to be able to use the specimens. So it's, first of all, we need the patients to work with us so that we can follow our vision of conducting the excellent work that will help everybody in the future.
As you go through the meeting and what's already been presented or what you know is going to yet be presented, is there something here that you have found extremely exciting?
Yes, and what I find exciting is the evolution of what has happened over the years, because you and I have met several years in a row and outside of the meetings, and to me it's really fascinating that every year we clearly see improvements. So for patients, they should continue to be hopeful that we're moving in the right direction.
Thank you very much. Dr. Edith Perez, Deputy Director at Large at the Mayo Clinic Cancer Center, Jacksonville, Florida.
Pleasure.